Welcome friends, I am Dr. S. Megala, Assistant Professor, Department of Microbiology, St.Joseph College of Arts and Science, Autonomous Cuddalore. Now I am going to talk about bacteriophage. Bacteriophages. Bacteriophage, also called phage or bacterial virus. It means viruses that infect bacteria. Bacteriophages were discovered by Frederick W. Dewarts in the year of 1915 and Felix de Herly in the year of 1917. De Herly coined the term of bacteriophage meaning bacterial eater. Next one, properties of bacteriophages. Bacteriophages also infect the archaea. Thousands of varieties of phages exist, each of which may infect only one type or a few types of bacteria or archaea. Like all viruses, phages are simple organisms that consist of a core of genetic materials. That means nucleic acids surrounded by a protein capsids. The nucleic acids may be either DNA or RNA and may be double-stranded or single-stranded. There are three basic structural form of phages. One is isohedral head with a tail. Next one, isohedral head without a tail and a filamentous form. Extracellular phage, that is phage particle existing separate from the bacterial host, are called mature or resting particles. Resting particles are sperm shaped less than 1 micron in length. Resting particle consists of entirely of DNA, that means genetic materials, and protein. Next one, life cycle of bacteriophages. During infection, a phage attaches to a bacterium and inserts its genetic material into the cell. After that, a phage usually follow one of two life cycles. It means lytic means virulent, another one lysogenic means temperate. Lytic phages take over the machinery of the cells to make phage components. Then they destroy or lyse the cell, releasing new phage particles. Lysogenic phages incorporate the nucleic acids into the chromosomes of the host cells and replicate with it as a unit without destroying the cell under certain conditions. Lysogenic phages can be induced to follow a lytic cycle. Other life cycles including pseudo-lysogeny and chronic infection also exist. In pseudo-lysogeny, a bacteriophage enters a cell but neither uses cell replication machinery of the host cells nor integrates into the host genomes. Pseudo-lysogeny occurs when a host cell faces the unfavorable growth conditions. It enables phage survival unit. Host growth condition became favorable again. In chronic infection, new phage particles are produced continuously over long periods of time but without killing the host cells. Next one, counting of phages. Bacteriophages are obligated intracellular parasites. They multiply inside a bacterium by making use of some or all of the host. It's a biosynthetic machinery. They enter the bacterial cells by landing on the cell wall and injecting the DNA into the bacterial cytoplasm. After entry, the page DNA acts as a template for production of page particles, causing lysis and death of the host cells. A bacteriophage particles cannot be seen by ordinary microscope. It can be seen only with electron microscope. Fortunately, we can use a technique very similar to the colony counting technique to count the page particles known as the plaque assay. Lytic pages are enumerated by this method. The plaque assay is employed to count and measure the infectivity level of the bacteriophages. It will also apply to measure and count the mammalian viruses as well. This assay is the most widely used technique for the isolation of virus and its purification and to optimize the viral titers. The basis of plaque assay is to measure the ability of a single infectious virus to form a plaque on plate containing respective bacterial culture. A plaque is developed as a part of infection of one cell by a single virus particles that is followed by the replication of that virus. And finally, the death of the cell, the newly replicated virus particles will later infect and then kill surrounding cells. Next, the double layer agar techniques. The double layer agar, hard agar serve as a base layer and mixture of a few page particles and a very large number of host cells in a soft agar form the upper overlay. When the plates are incubated, susceptible bacterial cells multiply rapidly and produce a bacterial lawn. When one page particle adsorbs to a host cells, penetrate the cells, replicates and release new page particles and infect other neighboring bacteria. This cycle is repeated until large number of bacteria have been destroyed. Cell produce single circular non-turbid area called plaques. The plaques became too large to be visible to our naked eye. Each plaque represents the lysis of page infected bacterial culture and can be designated as a plaque forming units. That means PFU. The number of page particles in the original stock page culture is determined by counting the number of plaques and multiplying this by the dilution factor. For a valid page count, the number of plaques per plate should not exceed 300 nor be less than 30. Plates showing greater than 300 PFU are too numerous to count. Plates showing fewer than 30 PFU are too few to count. This image showed that counting page. The page lysates and bacterial culture and soft agar. It's a gentle mixing and pouring into the content of a tube into the agar plate while still warm and liquid. And then solidification at the room temperature. Visualization of a page plate on the bacterial lawn. Next one, Properties of the page-infected bacterial culture. In the laboratory, usually infect a bacterial culture with a large number of page particles, and special techniques are needed to analyze the result. Genetic principles are nearly the same for all organisms, including human. So, in order to understand complex genetic process, genetic often used simple organism. Bacteriophage or the virus that infect bacteria, they are widely used for genetic analysis because generation time is short, haploid genome, large number can be propagated on the plate, easy to identify the plate. All genetic experiments start with mixing of phage with its host bacterium. The problem, after mixing, some bacteria are infected while others escape from infection, or some host cells are infected by more than one pages while other escapes. To calculate the efficiency of infection, two parameters can be used. First one, multiplicity of infection, MOI. Second one, fraction of uninfected cells. First one, multiplicity of infection. MOI is the rate of the virus to bacterial cell in an infectious fluid. MOI is the ratio between the concentration of phages and the number of bacterial cells allowed to infect. For example, if the value of MOI is 5, it means that the number of viruses are 5 times higher than that a host bacterial cell. If the MOI value is 1, it means only one page found per one bacterial cells to infect. Second one, the fraction of uninfected cells. The minimum fraction of cells that will escape infection can be calculated using following formula. PO equal to EMI. Next one, production of phage lysate. Phage multiply much more rapidly than bacteria. The number of progenesis released is depends on average of burst size. Burst size is defined as the average number of phage released per bacterium. The burst sizes may vary 10 to 100 for DNA viruses, and for RNA viruses it may up to 20,000. Be careful of choice of media, growth condition, and the time of infections of phage lysates. High concentration of phage can also be prepared by growth on solid media using an amount of phage such that all bacteria are ultimately infected and lysed. Next one, for example, if 10 to the power of 6 phage are the placed in soft agar with about 2 into 10 to the power 8 bacteria, after about 6 hours the soft agar layer will contain about 10 to the power of 11 cell phage. Such lysates is called a plate lysate. A plate lysate can be prepared from a single wire is stabbed into a plaque and then stirred in liquid soft agar containing bacteria. The soft agar is then poured on a plate. Certain kinetic parameters of the phage infection can be determined by studying an infected culture. A classic experiment is the one-step for growth curve. In this experiment, a culture is infected with an MOI of about 0.1. Phage antiserum is then added to inactivated any unadsorbed phage. The infection cells are diluted about 1000-fold into fresh warm medium. The inactivation of progeny page by the antibody. And at here various times aliquots are taken for plating the plaques. At first the number of plaques is constant because plaques are formed only by infected unlysed cells. This period is called the latent period. At a particular time after infection, the number of plaques increases. During this short time interval, the infected cells are lysing. When all the cells have lysed, the page concentration remains constant. The ratio of the page produced to the initial number of infective centers is the burst size, and the number of minutes before the increase in plaque number occur is the lysis time. A modification of this experiment enables one to determine the kinetics of page productions within the cell. In this procedure, chloroform and lysozymes are added at various times after infection. Chloroform destroy the cell membrane and lysozyme break down the peptidoglycan layer, which result in premature lysis of the infected cells. Thank you.